In this video, we are going to talk about the HMP shunt. It is a series of reactions that take place in the cytoplasm of many cells, such as the liver cells and the mammary gland cells, and it is important to produce many things. For example, it is the only way to produce ribose 5-phosphate. And the ribose 5-phosphate is very important to produce many useful things. For example, it is used in the production of nucleotides, DNA, RNA, and coenzymes. So this is the DNA and the nucleotide and some important coenzymes. And also the production of NADPH, which is important, and we're going to talk about the importance of NADPH later in that video. So before we start with the HMP shunt, we have an important thing to know about. So you know that glucose can be oxidized by two ways. The first one is the major pathways, and the second way is the minor pathways. So the major pathways, it includes glycolysis, and after finishing the glycolysis we would end up doing the oxidative decarboxylation, and after doing that we will get to the citric acid cycle. And in the minor pathways we have two ways of doing the minor pathways, and the first is Hexose monophosphate shunt, which is HMP shunt, and it is today's topic. And the second way of oxidizing the glucose by the minor pathways is the uronic acid pathway. And the difference between the major pathways and the minor pathways is in the production of ATP. So in the major pathways there is production of ATP, and as you see, there is production of ATP when we are oxidizing glucose by major pathways. But in the minor pathways there is no production of ATP, and that's a key difference between the major pathways and the minor pathways. So all of these are pathways to oxidize glucose, but the thing that you have to know is that all of them start with the same reaction. So let's take a look at this reaction. So now we have to start with glucose, and we are going to oxidize this glucose, but we said that the first reaction is similar, and we are going to use glucokinase, which is going to act on glucose and add a phosphate group on the 6th carbon. So we have to turn an ATP into ADP, and now we have a phosphate group out of this reaction, and this phosphate, the glucokinase is going to use this phosphate and attach it to the 6th carbon here in this glucose unit, and now we have a phosphate group on the 6th carbon, and it's called now glucose 6-phosphate because we have phosphate on the 6th carbon atom. Now after we form the glucose 6-phosphate, now we're going to get into the HMP shunt. So the first reaction, we are going to get rid of this hydrogen right here. So the enzyme is called glucose 6-phosphate dehydrogenase. So dehydrogenase means that it's going to get rid of one hydrogen atom, and this hydrogen atom is going to be carried by NADP plus, and now it is NADPH, and that's the first NADPH we formed in this HMP shunt. So after we got rid of this hydrogen atom, we will get another product, which is the 6-phosphoglucolectone. So do you know that the word lectone means that there is an ester group in a ring? So where is the ester group here in this ring? There is the ester group, which is incorporated in this ring. The next reaction, we are going to get rid of this word lectone. So to get rid of lectone, we have to get rid of the ester group. So how are we going to do that? We are going to break the bond between the oxygen, this oxygen here, and the 5th carbon atom here. So we are going to get rid of lectone by an enzyme called lactonase, and now this is the product. It's 6-phosphogluconate, not glucolectone, gluconate, because there is the ester group. It is now not ester group, it's a carboxyl group. So after we got 6-phosphogluconate, now we're going to do the same thing that we did in the first reaction here. We are going to get rid of one hydrogen atom, so the enzyme is called 6-phosphogluconate dehydrogenase because we are getting rid of hydrogen atom, and this hydrogen atom is going to be carried by NADP plus, and now it's turned into NADPH. And the product here, it is ribulose, not ribose, ribulose 5-phosphate. And the ribulose 5-phosphate is an isomer of ribose 5-phosphate, which we talked about first in this video. And isomers means that they have the same number of atoms, but they are different in structure. So here is the ribulose 5-phosphate and the ribose 5-phosphate, and they are isomers. So we are going to use an enzyme called isomerase, and it's called ribose 5-phosphate isomerase. So now after we produced NADPH in the HMP shunt, we have to know what is the importance of NADPH. So NADPH is used to produce reduced glutathione. So what is reduced glutathione? Now in this reaction we have 2 NADPH and it is turned into 2 NADP plus. So let's take a look at the oxidized glutathione. So the oxidized glutathione, we have the name, the shortcut for it is GSSG. So there is a bond between these two sulfur atoms. So you can say that this is a GS and this is a GS, and both of them are GSSG, and there is a bond between the two S, the two sulfur atoms. Let's get back to the NADPH. So where did this hydrogen go? It is now NADP plus. Okay, so this hydrogen is going to go to the oxidized glutathione to reduce it and form a reduced glutathione. We're going to break the bond between the two sulfur atoms, and we are going to get singles. We are going to get singles of GSH. So now we have 2 GSH out of this reaction. One here and one here. We're going to add an H to each one of these sulfur atoms. So what's the importance of reduced glutathione? So before we know the importance, we have to know an important thing that's going to help us to understand the importance of glutathione. So the H2O2, which is the peroxide, it is a lipid peroxidase. So when it's found in greater amounts, it can lead to cell membrane fragility. That's because 50% of the cell membrane is composed of lipids, and that's the cell membrane. 50% of it are lipids. Also, the H2O2 can lead to the oxidation of iron in the hemoglobin and red blood cells, and that can lead to anemia. So now comes the importance of reduced glutathione. So glutathione is going to remove the H2O2 out of your body. So now we have 2GSH, which we formed in the first reaction, in the previous reaction, and we have the peroxide H2O2, and now we're going to form oxidized glutathione, and we're going to turn the peroxide into a water molecule.